We're here at the 22nd CROI here in Seattle, Washington. We're here with Johnathan Li, and he is with Brigham and Women's Hospital in Boston. I'm Fred Scheich with IFARA from Portland, and we wanted to talk to you about viral rebound and a way of identifying that through the compartment and through the amount of virus on board. So let's talk to that.
Yeah. So as we all know, antiretroviral therapy has saved millions of lives, but our patients cannot get off of treatment, partly because HIV integrates into long-lived memory CD4 cells. And so they need lifelong treatment. One of the main priorities of the HIV field is to find strategies where we can control or eliminate the HIV reservoir so that our patients can go off therapy and achieve a long-term ART-free HIV remission. But in order to do that, we need to try to accelerate kind of the pace of the drug development process by looking for biomarkers. And that's, whether it's within the HIV field or outside of the HIV field, that is one of the key steps to be able to accelerate drug development. And even within the HIV field, the development of viral load as a biomarker greatly accelerated the progress of antiretroviral therapy development by taking trials that were extremely large and needed to achieve clinical endpoints and shortening it and making it much smaller because we were able to use viral load as a surrogate endpoint, as a biomarker.
And we had that actually came along. They were approving drugs based on the viral load, but they hadn't even approved the viral load. It's kind of a backward situation, so I hope we have a better situation with what you're talking about.
Absolutely. So what we're trying to do is to look for the viral load equivalent of drugs that are aiming for HIV remission now. And so what we did was that we took a look at 6 different AIDS clinical trials group studies that's already been done. These studies all included patients who stopped therapy. Some of them was for one reason or another, but generally to test therapeutic vaccines or other modalities. We took those patients who didn't receive any immunologic intervention, so placebo-arm patients. And we looked at their blood at the time when they stopped treatment, and we measured the HIV reservoir, the size of the HIV reservoir. And we looked to see which of the various aspects of the HIV reservoir best predicted when these individuals rebounded, the timing of the rebound. And we not only looked at the level of HIV DNA, but we also looked at the size of the active HIV reservoir. So when I say active, I mean they're actively transcribed. So cell-associated RNA, so RNA within a cell, but also the amount of residual viremia or low-level virus in the blood. And what we found was that this active viral reservoir, this cell-associated RNA and residual viremia, were associated with the timing of viral rebound, and that those who had the most delayed viral rebound also had started out with the lowest levels of this active reservoir. And so the hope is that that this is the beginning of finding biomarkers where we can use to more rapidly develop drugs in early phase studies and in vitro studies, and to determine which ones are the best ones to use into future clinical trials. So,
what biomarkers are you using? I mean, is it just viral load, or?
So right now, you're saying right now for looking at drugs for HIV remission?
Right.
So there is a myriad, a wide range of biomarkers people use. Everyone likes their own kind of pet biomarkers. Some people use HIV DNA, some people use HIV RNA, some people use HIV T cell activity, some people use viral outgrowth assay, and one of the problems is that we're in the dark about which of these biomarkers is best predictive of when the patients rebound.
You do all of them and then you show correlations, the best correlations?
That's what we're trying to do right now. So this is a start. We've started off with some of these measures of the HIV reservoir. We're going to move on to looking at immunologic biomarkers of viral rebound.
Or inflammation? Or
inflammation as well. That's exactly right.
I learned that there were like 19 or something like that.
There's a lot.
I thought there were like about half a dozen, and I'm learning now.
And they're adding more every day, it feels like.
Yeah.
So, but really what we need is to do more studies in this particular area, to do a comprehensive look at all of these biomarkers, and we're trying to do that with some of the existing samples. Samples are a little bit limited, and we're trying to also get a prospective study off the ground. We're designing a prospective study where we can test a whole range of biomarkers and be able to really pick the best ones that we can then use for future clinical studies.
But you can learn a lot from archived.
Oh, absolutely.
So it may not be, it won't be a trial per se because you have, picking here and there, but it will kind of guide you into developing a robust, very good endpoint trial. Are there any confounding variables that you might find, like flu-like or colds or anything that'll trigger, confound you, or will that be just a little bit of noise here and there?
I think that concurrent infections, I think it would probably be some baseline levels of low level of, one person has a virus here, one person has a virus here. I don't think that that's going to make a significant impact on the study at all. But there are things that, as you said, that could be confounders. For example, what we found was that individuals treated with certain antiretroviral therapy medications also tend to have delayed rebound, and that is the non-nucleoside reverse transcriptase class of medications, the NNRTIs, right? And the hypothesis is that these drugs have a much longer half-life. And so they have delayed clearance even after the patients stop treatment. And so they're actually probably have a prolonged exposure to drug even after up to maybe 2 or 3 weeks, even after they stop treatment. So it really speaks to the importance of assessing and taking into account the drugs that these patients are on when they stop therapy, when analyzing these trials, but also moving forward and designing new trials that patients are put on similar drugs, so that these pharmacokinetics confounders are not affecting your results.
How would you do? Just eliminate them from the trial, or would you just?
So there's various ways.
A sub-study with just those, or?
Yeah, so you could either limit your trial to patients only taking certain types of medications, like maybe integrase inhibitors or protease inhibitors, and not taking these NNRTI treated patients, or switching them for a limited time beforehand, before doing the interruption.
So where do you go next? You're building the trial and figure out what to include and what not to include, how to design the trial. Designing the trial is going to be really tricky. I
agree, and there's a lot of people kind of put together a good team. Some putting it through the ACE Clinical Trials group, looking at this problem and trying to figure out the best way of designing a prospective study to look for these biomarkers. At the same time, keeping in mind that top priority is patient safety. That taking patients off treatment is not trivial, and we don't consider it trivial. And
because we learned that was not a good thing to do. But if it's how long would you have patients off?
Yes. So in the past, when some of these studies have been done, I think oftentimes it hasn't been realized that it can have deleterious effects. Long-term viremia, having virus rebound for long periods of time, can be deleterious.
And that was the way we built the.
That's why the SMART study, right?
Yeah,
so,
exactly.
That's
not what you're going to do.
That's exactly right. That is not what we're going to do. The newer study designs.
Until you have rebound.
Patients, you're exactly on point. The patients will restart treatment as soon as the virus comes back.
And that should be sufficient to put them in a position of no loss of ground.
That's exactly right. That's exactly right. We're really trying to enhance safety, while at the same time making sure that these trials are scientifically rigorous.
Well, it sounds like you're heading in the right direction. I love it. It's just something that's, the sad thing is there's so many trials that need to be done. There's so many great concepts that need to be tested.
Yeah.
And I keep on saying this all the time. Every time you come up with an answer, you have 20 more questions. And I know that this one's going to trigger that. Or you say, gosh, we should have included this, or we should have done this or that. But like you say, the more people you bring into it with questioning, in a way that's constructive,
Yep.
to make sure that you design the trial the best way you can, because you can't change it. Well, you can, but not easily down the line. So I would say it's a challenge.
It's a challenge I'm looking forward to.
Jonathan, thank you very much for
Thank
being
you for
here.
having me.
Yeah.